that, we are going to move on to placenta and fetal sex and how that's associated with hypertensive disorder of pregnancy. So I thought I would give just a very brief tutorial, not knowing what people's backgrounds are around hypertensive disease of pregnancy. But hypertensive disease of pregnancy includes the condition of chronic hypertension, which is where women have blood pressure elevation going into pregnancy, gestational hypertension, where they were normotensive but have developed now blood pressure after 20 weeks of pregnancy. There's the preeclampsia, eclampsia spectrum that also includes HELP syndrome, where women develop not only hypertension, but some other end organ damage related to the disease process, including up to seizures or eclampsia. And then there are the women who came into pregnancy with chronic hypertension can develop superimposed preeclampsia on top of that. What I want to point out is for the EPOCH study, where we're really trying to understand what is the link between hypertensive disorder at pregnancy and later cardiovascular disease. We excluded those with hypertension before pregnancy, those with diabetes before pregnancy, because what we were trying to do was remove the pre-existing risk factors, because we really wanted to see what does pregnancy, how does that impact later cardiovascular disease. So I call this the new onset HDPs. So the gestational hypertension and preeclampsia is, when I refer to HDP in these studies, it's new onset HDP, so pregnancy specific. And as this audience is interested in and knows that this is associated with later cardiovascular disease risk, it's also been associated with increased kidney dysfunction, neurologic outcomes, so it's not just the heart. Preeclampsia does impact the full maternal body. And this pregnancy is a disease, a process for two individuals, not only the mom, but also the baby. And there's increasing evidence showing that these hypertensive disorders do impact fetal long-term health as well as the acute associations with growth restriction, stillbirth, and that type of process. So this is a disease that's really impacting two individuals, both in the acute setting as well as the long-term consequences. Risk factors for preeclampsia are listed here. You'll see that they're wide and varying, and I think this reflects that preeclampsia truly is a syndrome and not just one disease, even though the diagnosis is a consolidation at the end of pregnancy of a constellation of signs and symptoms. And what we understand about the pathophysiology of pregnancy is that it's complex and we fully don't understand it. So I think the importance of this is that there's clearly genetic factors, there's clearly maternal environmental factors, there's definitely immunologic factors, and all of these are impacting how the placenta forms and develops in early pregnancy and how it functions through pregnancy. So the placenta is really central to the pathophysiology of this disease process. And then the placenta is thought to then secrete material into the maternal circulation, which is then having the maternal systemic effects that we see as the syndrome of preeclampsia that presents with the proteinuria, the hypertension, other end organ complications, as well as HELP syndrome. And vascular endothelial dysfunction is a key central tenet of this process, as well as an enhanced inflammatory signature. So the way people typically think about preeclampsia is kind of in that longitudinal Abnormal placental development leads to a diseased placenta, which then impacts maternal endothelium, which leads to disease. But I think I like to also think about preeclampsia pathogenesis much more as an intersectional disease process, an intersection of two individuals, the fetus, i.e. placenta, since the placenta derives from fetal tissue, the embryo, and the mom. And so that you can get to preeclampsia both by having that abnormal placenta process. But I think there's also the maternal susceptibility, because some women who develop preeclampsia will have a histologically normal placenta. So there's the maternal susceptibility to probably a normal placenta, so the stress of pregnancy, normal pregnancy, as well as one of the other big risk factors is twins, large placentas, so just placental volume can lead to preeclampsia. So my favorite organ, the placenta. This is what it would look like depicted at the end of pregnancy in a cross-section. So I'm going to give you a quick Bio 101 on placental anatomy. So here, can you see the arrow? Okay. It's very sensitive. Okay, so this is the umbilical cord. This would be attached to the baby. The amniotic fluid would be surrounding in this area. You have your chorionic plate, and then the bulk of the placenta is this chorionic villus tree structure, or the parenchyma of the placenta. And this is the workhorse of the placenta. This is where transport is happening, and this is also where all the endocrine function of the placenta is occurring. The placenta is the largest endocrine organ that we had. And then the basal plate, or the decidua basalis, is where the placenta anchors into the maternal decidua, the altered lining of the uterus in pregnancy. And for the EPOCH study, we were very interested in looking both at the chorionic villi as well as the basal plate region of the placenta. They have very different functions, very different cellular makeup, and what I want to focus in is probably serving different contributions to the pathophysiology of preeclampsia. So the basal plate region is where the building blocks of the placenta, the cells, the trophoblasts, which were derived from the trophectoderm of the implanting embryo, proliferate at the anchoring ends of that chorionic villus tree and invade into the maternal tissue and remodel the maternal vasculature. So that basal plate region is really chimeric between the mom and the baby. And it is that remodeling of the maternal vasculature that allows for the required tremendous blood volume that needs to go to the uterus to support the growth of the baby. When those vasculature does not get remodeled, you have impaired perfusion leading to ischemic injury and thought to lead to the increased secretion of toxic factors into the maternal circulation. So this basal plate region is very important and thought to be part of the process in early pregnancy of developing pathogenesis of preeclampsia. The chorionic villi is probably more important in the later stages of preeclampsia, where this is where many of those factors are secreted into the maternal circulation to impact maternal physiology. So I've always thought in terms of thinking about syndrome and potential subtypes of preeclampsia. You may have women who have a strong basal plate signature versus those that may have a strong chorionic villi signature. And that actually knowing the signature of these different regions of the placenta, as well as knowing the signature of the mom in terms of her susceptibility, you probably need all three to really fully understand that intersection leading to preeclampsia. And what people often forget is that placenta does have sex. the placenta is derived from the embryo, so it is male or female, following the male or female of the fetus. People often think the placenta is mom, but it actually is not. It's male and female. And we know that when you look at the placenta, there are clear sex differences in the transcriptome, clear sex differences in its function. And so when we started to think about this placenta data that we have from EPOCH, we were very interested in wondering whether if the pregnancy was a male or a female pregnancy or a male or female placenta, would that impact the hypertensive disorder of pregnancy? So Dr. Shilpi Segal is a postdoc in my lab, and she set out to investigate both the sex-specific and placental regional differences in the placentas from new onset hypertensive disorder of pregnancy. So as a quick overview, which actually we got the preview during the turnover, this is the pregnancy cohort and midlife cohort of, that we performed here at Stanford. Like I said, we have blood and urine samples from third trimester, from delivery and postpartum, as well as our midlife cohort. And we also have placentas and cord blood from delivery. So when we got a placenta, we ended up taking biopsies both from the basal plate regions and from the chorionic villus regions of the placenta. We also took samples from the central portion of the placenta as well as the peripheral, because prior data, particularly actually Yoel Sadovsky, who's now here at Stanford, had shown that the transcriptome can be up to twofold difference in hypoxia-related genes when you look at peripheral versus central. So we wanted to make sure that we were covering that aspect. All the biopsies were processed within an hour of delivery, snap frozen, and then later RNA was isolated, underwent bulk RNA sequencing and analysis. For this analysis, in EPOCH, we did enroll twins, but for this analysis, we've limited it to singleton pregnancies. We excluded those with gestational diabetes and chorioamnionitis, and we ended up with a final cohort of 52 placentas, which nicely separated into 27 hypertensive disorders and 25 normotensive controls. And then we also had those 4 samples. So we had actually 208 different samples that underwent bulk RNA sequencing. When we look at the demographics of the women that these placentas were taken from, things that are different are to be expected. We know BMI is typically higher in women who have hypertensive disorder. The blood pressures were higher. That's a good thing. And lower gestational age and lower birth weight, all related to the disease process. Other factors were pretty well matched between the two groups. When we did initial PCA analysis of some of the most highly differentially expressed genes, what you can see on the left for the chorionic villus tree structures, there was a clear separation of the female placentas from the male placentas, showing that fetal sex is the dominant influence on the transcriptome. In the basal plate, you see that the female placentas are more towards the top of the PCA plot and males are more towards the bottom, but not nearly as much of a separation. But that makes sense because half this tissue is maternal. So the number of cells within the sample that would have had a sex difference is a lot less. So I'm going to focus on the chorionic villus tree component of this data as we move forward, thinking about sex differences. So what Shilpi did was then, since their sex was such the predominant factor, even more than hypertensive disorder, she looked at males only and looked at hypertensive disease versus normotensive, and female placentas, hypertensive versus normotensive. And these are the volcano plots of the differentially expressed genes. And the next question would be, well, how much of an overlap is there in those differentially expressed genes? And this is where the surprise came. There's actually very little overlap between what is differentially expressed in the placenta when you have hypertensive disorder, whether it's a male or a female pregnancy. And there was only 2 genes that overlapped. When we looked at the basal plate in the same way, fewer genes were showed differences, but again, the same theme, very little overlap between fetal male and female. So when you look at, she then took all the genes that were differentially regulated and did an unsupervised clustering, showing the heat maps here. And what you can see at the bottom is how the data separates or clusters the placentas, and actually did a fairly decent job of clustering controls around each other and hypertensive disorders around each other. But you can also clearly see here that it's not a binary, it's not clear disease, clear control, that there is also within the diseases what look like potential subtypes. And so we're going to be diving into that to have this molecular data help us understand better preeclampsia subtypes. And when you take all those genes and put it through pathway analysis, I'm showing you here the KEGG pathway analysis. When we look at the male placentas, the pathways that stand out as being upregulated in hypertensive disorder are immune-based, inflammatory-based genes. And the downregulation was cornified envelope formation. If anybody can help me with a little bit of insight on that, I'd be happy to hear it. But what's really interesting too is then when we go over to the females, the downregulated is cytokine, chemokine interaction. So inflammatory immune interactions is actually down-modulated in the female placentas. And what's up-modulated is RNA processing and translation of RNA. Again, very different biological phenotypes, whether it was male or female. So the conclusions from this is that the placental transcriptome in hypertensive disorders is actually very sex-dependent. And I think that that is something that we've missed thinking about. And I think this data strongly says we need to be paying attention to that. In the males, the alteration was much more immune-based, whether that is male, Y chromosome, a greater immune aspect, I don't know. And the females was more upregulation of RNA processing and translational pathways. Fetal sex needs to be considered in studies, I think, understanding hypertensive disorders in thinking about the pathophysiology. And what this really struck me is we need to be thinking now, we've been thinking about now what is the long-term impact of having it. But I think now we have another layer. We have to think about what is the long-term impact, whether it was a male or female pregnancy hypertensive disorder. So our future directions is we have a lot of data to do a lot deeper diving into understanding the relationships of the clinical variables with these molecular subtypes, taking into account not only the placentas, which can really, I think, anchor, because the placenta is driving these diseases, correlating it with the other omics data. We're working on, this was bulk RNA-seq, so working on deconvoluting the data so that we can see what cell types, maybe there's certain cell types within the placenta that are driving the sex-specific differences. And we're able to do that because we also have a subset of these placentas. We've completed a single nuclear multi-omic analysis on a small number of placentas, which will allow us to do the deconvolution of the larger data set. And I think we need to think about now going back to our midlife cohort and reassessing that data, keeping in mind the idea of fetal sex. Now, of course, that's complicated if you've had more than one pregnancy and they were of different sexes. So we'll see how the numbers fall out, but that's the way we've been thinking about this data. I have to acknowledge this EPOCH study was tremendous. It required a huge number of folks, a large number of teams and labs. We had basically 13 co-investigators, PIs, as part of this large grant. And it took a team of clinical research coordinate. It was a fabulous effort. Funding that I've had, I'm always grateful for, and I'm happy to take any questions.